My presentation today is titled Scientific and Technical Considerations Relevant to the Clinical Adoption of Human Germline Gene Editing. As compared with earlier endonucleases, CRISPR-Cas9 stands out in terms of its targetability, fidelity, malleability, and versatility. To accomplish its editing function, CRISPR-Cas9 needs to effectuate a double-strand break, or DSB for short, thereby allowing us to repair multi-base mutations by means of a mechanism known as homology-directed repair, or HDR for short. Unfortunately, DSB-dependent editing also gives rise to non-HDR DNA repair mechanisms, endogenous mechanisms known as non-homology end joining, or NHEJ for short, which gives rise to small stochastic on or off target insertions and deletions, also known as indels, as well as to large on-target deletions and complex rearrangements. For these reasons and others, David Liu from the Broad Institute and other investigators developed DSB-independent editing technologies that are designed to repair single-base mutations. These so-called base editors have the capacity to convert CG pairs to TA counterparts or AT pairs to GC counterparts. Which brings us to the main topic of today's discussion, that is to say, the editing of the human germline. The first question we may wish to ask ourselves is, what is the objective of germline editing? And the answer, of course, as was already stated, is to prevent disabling and life-truncating monogenic maladies, which is why we sometimes refer to the process as remedial germline editing. In so doing, we convert what is otherwise a function of chance to a function of choice, which gives rise to, admittedly, a lofty goal, that is, to rid humanity of, quote, the monogenic scourge, end quote. The second question we may wish to ask ourselves is, what universe are we seeking to tackle? Surely, we do not have in mind to edit the more than 10,000 human monogenic disorders currently listed in the online Mendelian inheritance in men. Instead, we are more likely to focus on edit-suitable maladies wherein peri or postnatal medical therapy is infeasible, maladies wherein peri or postnatal somatic editing is ineffective, maladies wherein pre-implantation genetic diagnosis, or PGD, is either inutile or gives rise to a rate-limited embryo complement. In short, we are likely to focus on target alleles that are highly penetrant, ideally singular, and most importantly, CRISPR accessible. The last question we may want to ask ourselves at this stage is, what is the state of the art? And the answer is nascent, which is to say, on the one hand, it is so near, but in another, it is so far. The first shot across the bow, if you will, in this field came about on April the 18th, 2015, when Professor Huang from Sun Yat-sen University published a paper in Protein Cell wherein he sought to edit human tripronuclear zygotes, that is to say, zygotes that are non-viable and therefore non-transferable and unlikely to ever give rise to a pregnancy. Professor Huang focused on the beta globin gene, which he edited and reported low editing efficiency, minimal editing specificity, and limited editing uniformity. Hardly a scientific breakthrough. Nevertheless, by dint of being the first, Nature did see fit to include this paper in its 10 People Who Mattered This Year and published the same on December 17, 2015. Less than 2 years later, the National Academy of Medicine released its report titled Human Genome Editing, which was focused on the field in general and did not specifically or exclusively focus on heritable human genome editing. Nevertheless, positions were taken. For example, the report suggested that research trials be limited to compelling studies of treating or preventing serious disease or disability, and only if there is a stringent oversight system able to limit uses to specified criteria. Later that year, the first consequential report on the editing of a human embryo saw press on the pages of Nature, courtesy of Professor Vitalipov from the Oregon Health and Sciences University. The report focused on the sarcomere gene, which when mutated gives rise to familial hypertrophic cardiomyopathy, a disease that is incurable but amenable to symptomatic care, and more often than not, will end up in sudden cardiac death. Editing of zygotes 18 hours post-fertilization, Professor Vitalipov reported editing efficiency of 67% and a mosaicism rate of 24%. Earlier editing, that is to say at the time of fertilization, increased the editing efficiency to 72% and reduced the mosaicism rate to 2.4%. In both cases, on-target indels were readily detected. Professor Vitalipov correctly concluded that zygotic editing at fertilization, 1, enhances editing efficiency and uniformity, if not specificity, and 2, that further optimization will be required if clinical application is to be entertained. Shortly after the publication of this paper, several critiques were published on the pages of Nature that questioned the validity of the results. The first question was, does the interhomolog repair claimed by Professor Vitalipov really holds up? Have CRISPR-mediated rearrangements, large deletions, and loss of heterozygosity been missed? And finally, has the hoped-for wild-type homozygosity been unequivocally documented? As mentioned earlier, late last year, a commission that represented the joint efforts of the National Academy of Medicine and Sciences and the Royal Society, co-chaired by Dame Davis and Professor Lifton, president of Rockefeller University, published its report titled Heritable Human Genome Editing. Whereas this report made multiple recommendations, the clinical recommendations thereof stated that no attempt should be made to, quote, establish a pregnancy with a human embryo that has undergone genome editing. Also, clinical use of heritable human genome editing should proceed incrementally. Further, clinical use of heritable human genome editing should be limited to serious monogenic diseases. And finally, clinical use of heritable human genome editing must guarantee impeccable editing efficiency, specificity, and uniformity, as well as euploidy and appropriate embryonic development. Not long after the release of the report, a critical paper saw press on the pages of Cell on December the 10th, 2020, emanating from the lab of Dieter Egli from Columbia University. In editing human embryos, Egli and his associates noted off-target effects of Cas9, which caused indels, but more importantly, they noted actual chromosome loss in the editing process, a rather substantial problem that would clearly have to be overcome. As we speak, 2 additional papers can be found on a preprint server that have yet to be peer-reviewed. But a review of those papers at this time suggests that they are unlikely to change the conclusions at this time with respect to the fact that heritable human genome editing is not quite yet ready for prime time. In summary then, embryo-based, clinically reliable heritable human genome editing is unlikely in the foreseeable future. Perhaps gamete-based heritable human genome editing, perhaps using cultured stem cell-derived eggs or sperm, may well prove the preferred route forward. At the present time, the stem cell-derived eggs and sperm have yet to be accomplished. Such has been accomplished in the rodent, but has yet to be accomplished in the human. And beyond that, one would have to document the feasibility of editing those gametes in the dish in a way that is, in fact, reliable, safe, and predictable. Thank you very much for the opportunity to contribute, and I will close here. Thank you very much.